Adam, grad student at the U of A and Catherine Maker's lab. I'll be talking to you today about the inhibition of MAVS signaling by PB1-F2 from H5N1 highly pathogenic avian influenza viruses in avian cells. So before we dive in, I want to give a brief background on a few key proteins, starting with PB1-F2. So PB1-F2 was first discovered in the +1 reading frame of PB1 in 2001. This was discovered using PR8, which is an H5N1 mouse-adapted strain. And depending on the strain, the PB1-F2s can differ in their sequence, length, and their contribution to the virulence of the virus. And so we compare some of the less virulent to the higher virulent strains, like PR8, like I said, it's mouse adapted, very low virulence in humans. The 2009 pandemic strain, if you recall, was highly infectious, but very low virulence in humans. The 1918 strain, very, very deadly. H5N1, also very deadly in humans, about a 60% fatality rate. These are both 90 amino acids long, and these are both 87. So in 2004, a couple highly virulent strains of H5N1 emerged, one in Thailand, D4AT, and one in Vietnam, VN1203. And we're also looking at a reverse genetics version of VN1203, and of course PR8. So if we look at some of the avian strains, you can see that the sequence does look pretty significantly conserved. But even a single amino acid change can confer a pretty significant difference in function of the protein. So if we look at PR8, we see a pretty significant amount of differences in the amino acid sequence. So a couple more key proteins, RIG-I. So RIG-I it recognizes the viral RNA of influenza. It will then upon detection, activate and tetramize, and these two card domains in the end here will get ubiquitinated by TRIM25, and then it will interact and activate MAVS at similar domains on the MAVS. This will lead to downstream signaling and type 1 interferon production. So this is where PB1-F2 comes in. PB1-F2 will inhibit this pathway by binding to MAVS and the mitochondria. Also of note is NS1, another influenza protein, and this is a very potent inhibitor and well-studied inhibitor of this pathway. So this serves as a really excellent control. So what we know so far is that activation of MAVS will lead to type 1 interferon production. PR8, PB1-F2 will inhibit this by binding directly to MAVS and to the mitochondria, disrupting the mitochondrial membrane potential, because this is very integral for MAVS-mediated signaling. And this was shown to occur in mammalian cells in that early 2001 paper, and we also showed it last year using chicken cells, in both cases using the PR8 strain. So how did we show this? So we used something called a dual reporter luciferase assay. So first we transfect cells with a plasmid containing the luciferase gene. Luciferase is a luminescent protein. It's what gives fireflies their glow. And this gene is under control of our chicken interferon beta promoter. So there's our type 1 interferon. And so we also co-transfect with a constitutively active version of luciferase. So this is a control. So this is from Renilla. It's a different host, but still luciferase. And this is always active regardless if this interferon pathway has been activated or not. And we also transfect with PB1-F2. This is flag tag, and this is the PR8 strain. And we need something to actually activate the pathway, so we can do this by co-transfecting with those two card domains that I talked about, or MAVS, or the full-length RIG-I, also co-transfected with a RIG-I ligand in order to be activated. And so once we quantify this in a luminometer, it looks something like this. So what we see here is that when we transfect with RIG-I, and pcDNA, which is empty vector, we get activation of the pathway. Empty vector alone will not activate the pathway. We also included the flag tag on its own with the rig-I as a control. And then when we add in PB1-F2, we see significant levels of inhibition. Also NS1, like I said, at excellent control. So now we want to investigate, are they actually binding to each other? And first we look at, are they co-localizing to the same part of the cell? So we do this using immunofluorescence. So if we go through these first top panels here, these are controls. So if we transfect DF1s, which is chicken cells, HeLa cells, human cells, with flag tag PB1-F2, and also mitotracker red, this will bind to mitochondria. And if we merge these two images, we see significant levels of co-localization. So PB1-F2 is co-localizing to the mitochondria. And then when we add in MAVS tagged with V5, we do see co-localization when we merge the two images. So we do know that PB1-F2 is co-localizing to the same part in the cell, but are they binding directly to each other? Because potentially PB1-F2, because it's co-localizing to the mitochondria, and it's binding to the mitochondria, potentially that's the mechanism, just direct binding to the mitochondria. So we investigate this using co-immunoprecipitation. So how this works is, if these are binding directly to each other, when we pull down MAVS using an anti-V5 bead, and if they are binding to each other, when we pull that down, the PB1-F2 will be pulled down with it. So I'll walk you through these first few lanes here. So in the first lane, we transfect with flag tag PB1-F2, and in the whole cell lysolate, we blot with anti-flag antibody, and we see the PB1-F2 show up, so that's good. In the second lane, we add just the V5 tagged MAVS. We pull it down with anti-V5 beads and then blot with anti-V5 antibody, and we see the MAVS show up here. And when we transfect with both the flag tag PB1-F2 and the V5 tag MAVS, and then pull down MAVS with the anti-V5 beads, and then blot with anti-flag, we see the PB1-F2 show up. So when we pull down MAVS with the beads, PB1-F2 is coming with it. So there is direct interaction. They are binding to each other. So what is not known so far is, will these PB1-F2s from the avian strains also inhibit chicken cells? And is the mechanism of action similar as it is with PR8? Are they co-localizing to the mitochondria? And then if so, are they binding directly to each other? So basically we recreate all these experiments, but this time including the 3 avian strains. And so we see here that whether the pathway is activated by the 2-CAR domains, MAVS, or rig-I, full-length rig-I with the rig-I ligand, in all cases we see significant levels of inhibition with all avian strains. So we know that the PB1-F2, regardless if it's a mammalian strain or an avian strain, it is inhibiting this interferon pathway in chicken cells. So next is to investigate, are they co-localizing to the mitochondria? And again, by performing that immunofluorescence experiment, we see in all cases, in all strains, when we merge them, they are co-localizing. So they are binding to the same part of the cell, which is the mitochondria. So we know that the avian strains will significantly inhibit the interferon production in avian cells, and we know that they will co-localize. So next is there direct binding to each other, just like there is with the PR8 strain? So again, doing that co-IP experiment, seeing if we can pull down PB1-F2 with MAVS. And then after that, we want to see if this can be replicated in duck embryonic fibroblasts. So if you recall, I mentioned that the VN1203 and reverse genetics version differ in their virulence in duck cells. So it would be really interesting to see if this PB1-F2, those 3 amino acid differences, is part of that difference in virulence. And if it is, that'd be very interesting to see, because it can show us which amino acids are most important. So yeah. And with that, I'd like to thank my supervisor, Catherine Magar, and all the members of our lab, my committee members, James Stafford and Troy Baldwin, Yana Zhao, who is a previous PhD student who started this project, helped me out with a lot of the project. She made a lot of the constructs. Daniel Evsey, another previous PhD student who helped out a lot with the co-amino precipitation experiments, and Robert Webster, who helped out with collaborating in infection experiments to get the RG VN1203 sequences. And with that, are there any questions?
All right. Hey, Adam, thanks for your presentation.
Thank you.
It looks like there's 42 people involved, and we need them to type in some type of question. I'm not going to lie, a lot of this stuff is kind of over my head, but it's interesting. I try to like practice some brain skills here. So I guess ideally, what's your end game? Like if you can, like is the end game to maybe try and establish some type of vaccine for people so that if they get H5N1, then they're not going to die, because you said the death rate's really high. Like what's the end game for your research?
So the research really in general just contributes to potential therapeutic targets of PB1-F2. So we're still not sure exactly like which amino acids are the most important. So doing these experiments and then doing them again in duck cells with the PB1-F2s that differ just in those 3 amino acids, but hugely in their pathogenicity. By investigating that, we can further narrow down which parts are most important. We know that the C terminus is the most important in general, that those last like 20 plus amino acids are the mitochondrial targeting sequence. So those are the most important. But yeah, continuing this research will just narrow down what is the most important parts of the protein, and it's a really good target because it's such an important virulence protein for influenza.
Dinesh has a question that says, which avian cells did you look at, and can you overexpress one and inhibit the other or each other?
We used DF1 cells. So DF1 is a immortalized chicken cell line. I'm not sure what you mean by overexpress one and inhibit the other, like overexpress.
I guess, yeah, so there's a clarification in the question there. It says, if you overexpress the PB1, can it totally inhibit the interferons?
No, so it's like at best, like even if you looked at NS1, which is an even more potent inhibitor of interferon, it's not a complete annihilation of the interferon response. PB1-F2 does have a couple other potential targets as well, also in the same region, which is of the cell, near the mitochondria, which is why we're interested if it's just in the area or if it's binding directly to MAVS. But yeah, overexpression will basically get us to the most amount of inhibition, but yeah, you can't completely get rid of all interferon response.
Right on. Thanks for your answers, Adam.